thank you all for coming out on this beautiful morning in, where am I? Washington. It's a long flight from the East Coast to the West Coast. And I want to thank Dr. Brown and the EvergreenHealth Center for inviting me. Plus, I want to thank all the sponsors who have contributed to making this a successful meeting this morning. What I'm going to talk about this morning is directly related to all the slides that Dr. Brown just put up there. And the reason for it is because as I go through this talk, you'll come to appreciate the common denominator for all those things that one can do about correcting and hoping to help themselves with MS are related to what we know as the gut microbiome. But that'll be right at the end of the talk. But in order to get there, you got to go through a lot of information first. What I'm going to cover during this next 45, well, probably 50 minutes is what is the microbiome, what is it comprised of, and then I will talk a little about immunology, because obviously there's a correlation between what goes on in our gut and our immune system and how that's related to multiple sclerosis. And then I'll talk about diet and how diet has an effect on this organ known as the gut microbiome, and I am calling it an organ. It's no different than our heart and our lungs and our kidneys. The only difference is that it was only in the last 15 years that it was discovered to be an organ of our body. So even though most physicians and nurse practitioners and nurses don't really learn much about it, the speed is picking up, and you'll understand why we don't know much about it, because it's probably the most complicated organ in our body, as well as in all animals and plants. And then I'll finish talking up at the end about risk factors and how they're all related to the microbiome. So let's first talk about ecosystems, okay? Because that's what this morning's discussion is going to be about, is ecosystems. And this is a very simplistic diagram of the Brazilian rainforest. And why do I put that up there? Because up until we've come to understand the gut microbiome, the Brazilian rainforest is probably considered the most complex ecosystem that we're aware of. But interestingly enough, our bodies have our own ecosystems, okay? And the ecosystems in our body are related to the mucosal surfaces. And what you see in this slide is that there's a number of mucosal surfaces, and each one of those mucosal surfaces has a microbiome. What is the microbiome? The microbiome is the interaction of the host, all the genetic information in the host, with the genetic information of those microorganisms that inhabit that space. So it's a combination of the host's genes as well as the microorganism's genes, and together, that is the microbiome. The microbiota, those are the organisms that comprise this space, okay? And as you can see in this slide, there's a number of spaces. There is the gut, which I will spend most of the time, if not all the time, this morning talking about, because it is the largest of the microbiomes. The skin, at least in the areas of the skin that have a mucosal surface, is a microbiome. The mouth is a microbiome. The vaginal canal is a microbiome. So basically, our eye, our lungs, any mucosal surface in our body has its own microbiome, and they all are different. They're different because they're all comprised and they are inhabited by different microorganisms. And what it shows here is that the GI tract, and that is the gut microbiome, is probably the most studied to date. And when I say most studied, you have to understand this is a new science. So there's going to be a lot of things that I bring up, and you're going to have way more questions than I can possibly answer, because our information in this is only fairly recent. So the gut is the largest ecosystem that we're aware of. And the reason for it is because within our gut, we have many, many bacteria. We have many viruses, parasites, a few funguses. There's all kinds of things that are down there. And they all live happily together, right? So we're filled with these bacteria, with these microorganisms, and yet we never get sick from it. But if things get out of balance, things do change. And that's what I'm going to be talking about, is when things go out of balance and what happens. Anyways, that is known as the gut microbiome. So back in the mid-1800s, of course, the late 1800s, there was a famous French microbiologist that many of you have heard of, which is Louis Pasteur. And Pasteur made this comment that life would not long remain possible in the absence of microbes. Now, I don't know if Kasper appreciated the complexity of the gut microbiome, but certainly as a microbiologist, he was a visionary because the statement is absolutely true. We cannot exist in the absence of all these microorganisms within us. They can't live without us, and we can't live without them. It's what is known as the symbiotic relationship. So they're doing what they need to do, and we respond to them. So what is it comprised of? Well, we have, as humans, depending on what data you want to believe, anywhere from 25 to 28,000 genes, okay? That's what sort of the number is these days. The bacteria, and I'm just going to talk about the bacteria alone, within us has anywhere from 2 million to 20 million genes. So we're outpowered approximately 100, maybe 1,000 to 1 by the genetic information of the bacteria within us than ourselves. And you have to remember, this is all alien material. These bacteria are not us. They're somebody else. They're Martians that live within us. They're our friends, even though they're aliens, and they reside within us. How about cell-wise? Well, this is even more interesting. We have approximately 10 trillion cells in our body. The bacteria alone in our gut, and I'm just talking about our gut, numbers 100 trillion. So we have at least 10 times more bacteria cells within us than we have cells in our own body. So we're outgunned by these microorganisms genetically as well as number-wise. Does this mean anything? Well, these bacteria are not there. They're not dead. They're not dormant. They're just, not just sitting there. They divide. Healthy bacteria divide anywhere from 1 to every 3 hours. So they're rapidly dividing. So it may be, 5 turnovers in a 24-hour period. They have all these genes. What do these genes do? Well, they produce products. They produce metabolites because it helps the bacteria grow. And these metabolites don't just sit in the lumen of the gut and then we excrete them. No, they pass through the lumen of the gut into our body. And it's estimated that each one of us in this room has approximately 500,000 bacterial metabolites within us. So we're basically a walking bag of bacteria. Now, even more interesting is that these bacteria are millions of years old, okay? We as humans are about 125,000 years old as a species. The bacteria within us, as I said, are millions of years old. In fact, we have a certain bacteria within us, a phyla bacteria. Remember back to high school biology, kingdom, phylum, class, order, family, genus, species. So phylum is right up at the top, so there's lots of bacteria, species under that. We have a phyla within us that are called archaea, and they are 3 billion years old. So we carry around bacteria that are 3 billion years old. So it becomes not only a scientific question, but it becomes a philosophic question, is what came first? Did the bacteria come first, obviously, and that we adapted to the bacteria, or did the bacteria adapt us for their liking? I don't know. That's a question that I certainly can't answer. But it's just something to think about, that these bacteria have been around, and we grew around them. Anyways, there's 2 major phyla within the gut that are of concern and interest. One is called firmicutes, which are gram-positive aerobes, and the other one is called Bacteroides, which are gram-negative anaerobes. And remember, these are phyla, so there's hundreds of species underneath them. How many hundreds? It's estimated, we don't know, but it's estimated that we have anywhere from 2,000 to 5,000 different species of bacteria within us. And the reason we don't know, because we can only culture about a third of them. So anywhere from 60 to 65% of bacteria, we have no idea what they are. And this is as of August 25th, 2017. So even though science is clever, we aren't that clever yet. And that's why I said, right from the start, this is a very complex organ that's doing lots of things that we don't know very much about. Anyways, the functions of the microbiome are listed here. There's importance in competing with pathogens, so we're exposed to nasty stuff all the day. The bacteria within us defend us against us. They actually defend themselves, is what they're doing, because it's a closed luminal space, and the pathogens get in there, but the bacteria are able to ward it off, and that's one way that we remain healthy. They also are important in maintaining the integrity of the epithelial barrier in the gut, the lumen of the gut, otherwise it'll be leaking all the time. And we know that there are certain conditions, and it may turn out that MS is one of those conditions, that's associated with what we a leaky gut. And I'm sure a lot of you have heard about leaky gut. And what that is due to increased permeability of the lumen of the gut. And the bacteria in the gut in particular are responsible for maintaining that integrity. There's very basic physiologic functions of the microbiome, such as digesting foods that I'll talk about in a little bit. But most importantly, at least I'm an immunologist, from my perspective, one of the most important things that the microbiome does is it maintains our immune system. And I'll explain to you why that is in just a bit. So here's just a sort of a diagram of what's going on with the microbiome. So here's the gut lumen, here's the bacteria, et cetera, in it. Here's the wall of the gut, and then on the other side you have the immune cells that I'll be talking about. Anyways, the host responds to the bacteria produces these metabolites, and together this idea of coevolution happens, and we have evolved simultaneously with the bacteria within us. And again, that is what the microbiome is all about. Now, we've come to appreciate in the past few years that there is a continuous discussion between the gut and the brain, okay? And I'm hoping that you'll walk away from this discussion this morning and say, geez, you know, that gut is really doing a lot, because I happen to think it's the second brain. As a matter of fact, I've come to appreciate that may be our first brain, okay, and our brain responds to it. And why is that? And that's because of this gut-brain axis. So as the gut and the bacteria in the gut are replicating down here, they produce all these metabolites that I told you about. These metabolites pass through the lumen of the gut, and they get into our system, they go up to the brain. And when that happens, the brain responds by producing a whole wide range of molecules, vasoactive peptides, satiation molecules. It's a pretty long list of molecules. And those go into our circulation, and they control the physiology of the gut. They control the motility of the gut, and the gut being able to move forward. Because you all know that when you're hungry, your stomach gurgles. It's after your intestine that's gurgling. And then after you eat, it goes away. Well, why is that? Because the bacteria have been fed. They release metabolites because they're happy now. Those metabolites go up to your brain and release the satiation molecules. Those satiation molecules go down to your gut, and they slow down the motility of the gut, because when the motility of the gut slows down, it changes the ability of the bacteria to replicate. So there's this continuous discussion, 24/7, 365, between the bacteria in your gut and your brain. And how is that going to relate to all the things that Dr. Brown was talking about? Because diet, in particular, as well as a number of other things, affect those bacteria that are in the gut, and by doing that, they affect the transmission of the metabolites to the brain and so forth. So why am I talking to you about this as an immunologist? Well, the largest immune organ in our body is our gut. 80% of the immune cells in our body are in our gut. So it's the reservoir of many of the things that drive the immune system. It's the reservoir for many of the cells that regulate the immune system. And there's a constant back and forth in the balance between those factors that drive inflammation and those factors that regulate inflammation. The gut is an enormous organ. It's 200 square meters, which is probably about half the size of this room, and that's just the absorption space. And that's what's filled with all these bacteria. But the bacteria themselves don't live in the entire gut. Your stomach is basically sterile. The first part of your small intestine, the ileum, is, not the ileum, but the distal part of the gut, the small intestine, that's where you start to see the bacteria, and primarily in the colon. That's where most of the bacteria live. We know that if we raise germ-free mice, they're unable to have an effective immune response. Why? What do we see? Well, first of all, there's smaller and abnormal lymph nodes in and around the gut called the mesenteric lymph nodes. There's fewer cells that are integral to the lumen that are part of the immune system called the lamina propria. So there's fewer of those cells. The cells that are there, the innate cells, remember there's two compartments to the immune system. There's the innate compartment, there's the adaptive compartment. The innate compartment is what we're born with that has these molecules on the surface called pattern recognition molecules, TLRs are the major one. They're the molecules that when you're born, when you first get colonized by bacteria, whatever it may be, because you haven't developed memory to your immune system, they protect us, okay? And they're called pattern recognition molecules, or toll-like receptors, the TLRs. Anyways, germ-free mice have an insufficiency of those. They have reduced lymphocyte numbers. And probably most importantly is that they have a reduction of these very important pro-inflammatory cells that are called Th17 cells that produce a molecule called IL-17, which, as of today, we believe is the primary driver in multiple sclerosis, okay? So germ-free animals cannot mount an immune response, and you can't induce an experimental condition that I'll talk about in just a bit called EAE in these animals because they can't mount an inflammatory response. So once those metabolites get out into the circulation, they activate the immune system, and I won't go through this in obviously any deep detail. So you have these cells, which are called APCs. Those are the innate cells that I was telling you about, and they have on the surface, as you can see here, those molecules that have the pattern recognition, et cetera, et cetera. So that's PRRs, or the pattern recognition molecules. They pick up whatever it may be. In this case, it will be a microbial antigen through a series of co-stimulatory molecules. And the reason these are important, and I won't go into that, is because there's actually a number of therapeutics that are coming down the pipeline for treating MS that are actually directed at that interaction between the two. They activate what are known as T cells. T cells are the compartment of the immune system, the adaptive immune system, that's responsible for something called cell-mediated immunity. Cell-mediated immunity is that when the lymphocyte goes out and attacks something, it attacks either a bacteria or a virus or a malignant cell. And those are usually done by T cells. T cells come in different flavors. They come as what we call Th1 cells, which are very pro-inflammatory molecules. For many years, we felt that Th1 cells were the cells that were driving multiple sclerosis. They also can become these Th17 cells. These are now the cells that we currently believe are most involved probably in the progression of MS, the pathogenesis of MS, because when we look in the brains of MS patients, the lesions themselves are loaded with IL-17 and Th17 cells. Now down here at the bottom are the Th2 cells, and Th2 cells are also cells that can drive the immune system, but in the situation of MS, they actually are protective. They're beneficial. And we have therapies now and have had therapies that polarize the immune system to a Th2. That was the understanding behind Copaxone. That was the understanding behind interferon, is that it would drive the immune system to a Th2 cell, and those Th2 cells would suppress the Th1s. And that was good up until we came to understand about these Th17 cells and this cell, the regulatory cell, which is probably the most important cell, the immune cell in our body, involved in regulating the immune system. And I'll just tell you right now, right up front, that what we believe as of today, that the major deficit in MS is that the regulatory T cells are there, but they don't function. They're insufficient in their function in those with MS. And the whole idea is to try and get those regulatory cells back and running and stable so that they help those with MS, okay? Why is this related to the gut? Well, the Th17 cells, those are the cells that live primarily in and around the distal ileum, the distal part of the small intestine. That's their home in the mesenteric lymph nodes that are there. The regulatory T cells, where do they live? They live in and around the colon, in not only the mesenteric lymph nodes, but in another small lymphoid organ called Peyer's patches, okay? So the major business that goes on in MS is between Th17 that's driving the disease and regulatory T cells, or B cells, both, that are controlling the disease. And the home for all these cells is in and around the gut. And that's why the gut microbiome is of increasing interest and importance. As I said, we've been around for 135,000 years or so as a species, but if you go all the way back to, worms and move up the evolutionary scale, each one of these has a microbiome. And it's been essential for their survival that they have this microbiome. And it's not just limited to the animal kingdom requires a microbiome, except the microbiome for plants is in the soil, in the roots, okay? But again, the same thing happens as you have all these bacteria in and around the roots of plants, in and around the gut of animals, and these bacteria are absolutely essential in the well-being of all living creatures. And just as an aside, it's estimated that the mass of bacteria in our world, our globe, is approximately 50 times 10 to the 30th, okay? It's the largest biomass of all living material. All the animal life, all the plant life together doesn't even come close to the biomass of bacteria. So not only are we a walking bag of bacteria, but the Earth is a walking sphere of bacteria that spins around the universe. So this is about how it's organized. As I said, your stomach is pretty sterile for the most part. Duodenum, that was the word that I was looking for that I blanked out on for a moment. And the duodenum leads into the jejunum, and that's usually pretty sterile. And once you get down to the ileum, right down here, that's when you start seeing the bacteria, and that's where you primarily see the IL-17 cells is in and around here. And then when you get into the colon, this is where you see all the bacteria, okay? And this is where, of the 100 trillion, the predominance of them live is in and around the colon. What do the bacteria do? Well, pretty straightforward. At least that's what we thought up until 5 years ago, and now we realize that the list for this is far more complicated. But in the small intestine, it's usually associated with carbohydrate uptake. Many of the immune cells in our body are in the small intestine, and it is involved in the maintaining of homeostasis, primarily through the immune system. The large intestine is involved in nutritional uptake. It gets rid of nasty metabolites that are being produced, and most importantly, from an immune standpoint, it ferments products that result in the development, or the production, I should say, of something that's called short-chain fatty acids. And short-chain fatty acids are the drivers of regulatory T cells. So it's through fermentation of green leafy vegetables for the most part, and that's why Dr. Brown said plants, eat plants, and that's why, is because what you're doing is you're stimulating the production of short-chain fatty acids, and I'll get into that in just a little bit. If you look at the composition in the human microbiome, you can see that, at least this is in the Western world, that there's a predominance of firmicutes. Firmicutes are those gram-positive aerobes that I was telling you about, and the other one is the bacteroidetes, which are the gram-negative anaerobes. Now, why is this important? And this is a very general comment. It's very general, is that the firmicutes are, for the most part, associated with the induction of those Th17 cells. But again, under the firmicutes of the phyla, there's hundreds of different species. Some drive the IL-17 response, some don't do that. So it's not all firmicutes. Not all of them are inflammatory driving necessarily. Now, bacteroidetes, as I said, are the gram-negative anaerobes. They're involved in the induction of those regulatory cells. So that's where the balance of the immune system is going, is between the balance of these two phyla within us. How is this related to our physiology? And I always find this pretty fascinating. So this was a study that was done a few years ago. So what they were doing is that they were looking at kids in Africa, okay, and this is out in the middle of nowhere, and it's essentially a vegan diet. Their source of protein, whether you like it or not, actually it's probably gourmet, is termites, okay? That's what their protein source is. If you go to those fancy restaurants, they'll charge $100 for it. And they'll give you 3 on a plate. Then you say, can I have some pizza? Anyways, they compared that to kids in the EU that eat the healthy diet, the same healthy diet that our kids eat, you know, lots of milk products and yogurt and cereal, et cetera, et cetera. And they looked at the colonization, okay? And you can see here that the kids in the EU, that's the EU, they had mostly firmicutes. And the kids in Africa, and this is age matched, have mostly bacteroidetes. Well, why is that important? In these these areas of Africa, which was similar to what it was like before World War II, autoimmune diseases are essentially unknown. Since World War II and the change in the diet and the globalization, so that they now drink Coca-Cola and eat yogurt and probably red meat and all that other kind of stuff, that pattern has changed. And now these kids now have, their gut flora looks about the same as the EU. How is this important as far as health is concerned? Well, here if you look at these mice, so there's one big fatty and one skinny mouse. These mice were germ-free mice. They'd never seen bacteria before. They're born germ-free. They're maintained germ-free. The mouse on the left, the big fatty one, was colonized with firmicutes, and the mouse on the right was colonized with Bacteroidetes. So these bacteria, not only are they involved in the immune system, but they're also involved in our health and well-being as far as obesity is concerned, because the mouse on the left is obviously grossly obese and has type 1 diabetes. So these bacteria, you start to get a flavor. These bacteria aren't just hanging out, they're doing something. Going back to the question that Dr. Brown raised, which is what is dysbiosis? Dysbiosis is when we're exposed to anything that changes the balance of those bacteria in our gut. And there's lots of things, and I'm not going to go through them because it's a full lecture within itself. But there's not very little we can do about host genetics, et cetera, et cetera. Medical practice, antibiotic use, especially antibiotic use in little kids before the age of 3 is a real factor. Kids before the age of 3 that are exposed to repeated oral or any kind of antibiotics have a much higher incidence of childhood autoimmune disease, eczema, in particular, asthma. And it depends on how you're born. If you're born vaginally, then you have the likelihood of developing a normal colonization pattern. If you're born by cesarean, your initial colonization is in the hospital and not your mother's vaginal canal, not the birth canal. So there's a total different setup. Kids that are born vaginally have a lower incidence of childhood autoimmune diseases than kids that are born by cesarean section. These are just observations, epidemiologic observations that have been made over the years. Now, we've come to appreciate also that there's other factors that are involved, and hygiene is a big factor. And if you look at this map, you can see that the prevalence of MS is generally higher as you go into the northern latitudes. It's becoming less and less so as globalization has occurred, so the accuracy of this is changing, and the same thing with type 1 diabetes. And what we have come to appreciate is that as we have cleaned up our environment, okay, remember, we're 135,000 years old, and it's only been in the last 100 years that we've decided that we should have clean hands, and it's only in the last 10 years that you walk into the grocery store and there's peripheral hand cleaner everywhere you go. We're basically dirty creatures that in 100 years since technology and industrialization has come in, that we've appreciated that we can lower the incidence of infectious diseases by just getting cleaner. That's the major anti-infective thing that we can do as humans, is have clean hands. You think it's penicillin and all that kind of, it's not. It's been shown clearly that it's related to hygiene. So we've gotten cleaner, and as we've gotten cleaner and the incidence of infectious diseases has fallen, the incidence of autoimmune diseases, including MS, as well as the other ones, has risen drastically. This is known as the hygiene hypothesis, and it's just something to think about, that we do live clean lives, and maybe they're too clean. These are just a few of the conditions, and I'm sorry, this is a little bit of an older slide. This list is much longer now on the left side, and I just throw that up there to show that basically the dysbiosis is what leads to this imbalance, that leads to these experimental conditions. Everything that I'm talking about this morning, for the most part, is at the experimental stage. We are just on the verge of being able to move into studying humans, again, because of the incredible complexity of this. Does the microbiome change over time? And the answer is yes. We start initially with a relatively sterile gut, not completely sterile, but it's pretty close to that. And we have low diversity. What is diversity? Diversity is the range of bacteria. As I said, we each have about 2 as an adult. We have anywhere from 2,000 to 5,000 different species. But when we're born, as I said, if you're born vaginally, it's mostly staph and strep. That expands over about a 3-year period. And during that time, from 0 to 3, you develop a mature colonization pattern. Why does that happen? Well, because you start, instead of drinking mama's milk, you start eating solid foods, and you're suddenly outside, and you're eating the dirt, and you're licking the dog, and the dog licks you, and so on and so forth. And that leads to the maturation of the microflora in the gut. Now, something really interesting that we don't understand happens to about between 3 and 5 years of age is that those bacteria don't change any longer. They become set. And that's what you've got pretty much for the rest of your life, okay? So the problem for kids who are treated repeatedly with antibiotics at an early age is every time they get treated with antibiotics, wherever their colonization has matured to, it backpedals, because one of the things that antibiotics do, not only get rid of the bacteria in their ears, but it also changes the bacteria in their gut. So they backpedal. And you stop the antibiotics after 10 days, and guess what happens? It takes about a month and a half to get back to where you were. And you will get back to where. But if you get treated repeatedly, you never get to the point that a kid who doesn't get treated with antibiotics gets to between 3 and 5 years old. Because at 3 and 5 years old, the immune system has become tolerized to those bacteria in the gut. And as I said, the gut flora at that point remains pretty stable until we get into older ages. What happens as we get older is that the bacteria colonization pattern does change. So if we're living in the Western world, which we all do, instead of being predominantly firmicutes, which is the Th17 driving cells, it becomes more of bacteroidetes, which is the regulatory cells. How is this related to disease? And I'm just throwing this out as speculation. Well, MS is a disease of young people. Autoimmune diseases, for the most part, are diseases of young people, right? So you've got these bacteria driving these IL-17 cells, which is driving the disease process. What happens as you get older? We don't get autoimmune diseases for the most part. What do we get? Well, we get cancer. Why do we get cancer? Well, maybe it's related to the fact that there's bacteroidetes in the gut driving regulatory cells, and you end up over-regulating the immune system so that you're unable to clear those malignant cells that arise all the time. So maybe our whole disease pattern, shifting from early on inflammatory diseases to later on malignant diseases, is related to what's going on in the gut. And this has become a very hot topic in the world of oncology now, because they're actually catching on to this and starting to think, well, maybe this is what the inherent problem is of aging and why we become susceptible to some diseases, certain diseases. So these are the factors, as Dr. Brown pointed out, that not only affect MS, but they directly affect the microbiome. So what happens is that we have these bacteria in the gut, and if we get treated with antibiotics or diet for a long enough period of time, and I'll explain to you in just a second how long a period of time that is, we get changes. So we see fewer bacteria of pink than we do of white, and you can see the whites generally go up in response to the pinks going down because this is a lumen, so it's always trying to balance itself. So if we lose one, the other one goes up. If you use that one, the other one goes down, the other one goes up. So it's back and forth. And when you change the diet or stop the antibiotics, it pops up back to where it was before. And that's exactly what should happen. So when you're on antibiotics as an adult, your flora changes for a period of time, and you all know that there are certainly antibiotics that cause a lot of GI distress, and that's because it has caused a shift in the bacteria. And that when you stop the bacteria, it goes back to that pattern that was before. So this is just a normal situation that swings back and forth. So the question then becomes, now, are there ways that these commensal bacteria, commensal means that there are these bacteria that live within us, that's why they're called commensal, are they involved in the control of disease process, in particular for this audience, multiple sclerosis? So let me talk, as I said, almost all this stuff is done experimentally. I'll talk a little bit about human work, a little bit that's been done, but this is mostly experimental at this point. So what we use is this model that's called EAE, experimental allergic encephalomyelitis, okay, or EAE. There's basically three different forms. There's active EAE, in which we take mice, certain strains of mice, we treat them with myelin-basic protein, which you all know is a component of the demyelinated MS, and we give that along with an adjuvant, and about anywhere from 16 to 20 days later, they develop a neurologic syndrome that's with leg paralysis, upper or forearm paralysis, et cetera, and it looks a lot like MS. It's mostly a spinal cord disease rather than a brain disease. The other way that we can do is we can take cells from animals that we induce the EAE in, immune cells, and we can passively transfer those into normal mice, and those inflammatory cells are able to use an EAE model. And then there's a third model, which is really intriguing, is where we take mice and we genetically engineer them so that they become spontaneous, they develop spontaneous EAE. All right, so these are the three different models of EAE that we use. The ones that I'll be talking about is primarily active EAE. Now, what we've learned from EAE is this, and I'm not going to obviously go through this, but we know that the pathogenesis of the MS, and that's what this slide is all about, is a remarkably complex product. It involves a whole bunch of different cells, all kinds of different cytokines that are produced. And let me just give a more rational and simplistic idea of what's going on during the pathogenesis of MS. So somewhere out in the periphery, and I happen to believe that the periphery is your gut, because that's where all the immune cells are, you have those antigen-presenting cells, the innate cells that I was telling you about right at the beginning, interacting with T cells, for example. And when that happens, the T cell becomes activated through those co-stimulatory molecules, becomes those Th17 cells, they proliferate, and then they migrate. They get into the bloodstream. As you can see here, they're now in the bloodstream, and they have factors on their surface that allow them to attach to the wall of the blood vessel in the brain. And when they attach to the wall of the blood vessel, they're able to open it up because they secrete very specific enzymes. Those enzymes cause a rent in the capillary wall, and the inflammatory cells get inside the brain. That rent is the same tear in the wall that when you have an MRI scan and you get infused with dye, it's through that tear that the dye gets into the brain that causes what is known as an enhancing lesion. And those of you with MS have heard all about enhancing lesions. And what that tells your doctor is that you've had an immune attack on your nervous system sometime in the last 4 weeks, 6 weeks, okay? But it's a tear, it's a bruise, and it heals up, and that's why after 4 to 6 weeks or so, that lesion no longer enhances because the dye can't get through the wall of the blood vessel. But anyways, once the cell gets inside the tissue of the brain called the parenchyma, it gets reactivated. And when it gets reactivated, it's by one of the cells that are similar to the ones on the outside, those innate cells, they're called microglial cells. On the inside, it could be another cell, it's called a dendritic cell. And those cells reactivate that inflammatory cell, and then that inflammatory cell begins to secrete molecules, in particular something called nitric oxide, and that was what causes the demyelination of the nerve. So this is a relatively straightforward cartoon of what's happening during the pathogenesis of MS. And the question is, how is this related to the gut microbiome? Oh, and then you have the regulatory cells, which are also activated at the same time as the inflammatory cells. And the idea of the regulatory cells is that they go into the brain or wherever the inflammatory cells are located, and they cool it down through a bunch of different ways that they can do that. So how is this all related, what I just told you, to the gut? Let's go back to the germ-free animals, okay? Those germ-free animals, I told you before, have never seen a bacterium. And as you can see in this figure very nicely, that germ-free animals, here's the disability score, very similar to the same disability score you get that your physician will give you. In humans, it's a 0 to 10 scale. In mice, it's 0 to 5. And you can see that the germ-free animals essentially don't get any disease. They're the open boxes. Here's a normal animal. Okay? They do get EAE. As I told you, it's usually between 16 and 20 days is the peak of disease. And this is associated with, as I said, limb paralysis, whatever. It's a monophasic disease, so it's not like MS that comes back over and over again. It's monophasic, it just happens once, but it's the best model that we have. And that is associated down here, as you can see, with that Th1 cytokine that I was telling you about several slides ago. There's a reduction of gamma interferon in the germ-free animals, and there's a reduction of that IL-17 molecule in the germ-free animals. Interestingly enough, there's an increase in the regulatory cells. That's what this great column is the regulatory cells associated with this marker in these germ-free animals. So the germ-free animals have increased regulation, but they have decreased inflammation. Now, if you take that animal and you colonize it with a member of the firmicutes, that family of bacteria that drives the Th17, but you just need one species, you don't need 2,000, just need one species, you colonize these germ-free animals, and look what happens. The animals now can develop EAE. So you need these gut bacteria to drive an inflammatory response that gives rise to the disease process in EAE. Here's another example. This is that spontaneous model that I was telling you about. So these are animals that they're known as transgenic animals. So here's the germ-free animals that, well, here's the wild-type animals that develop spontaneous EAE. And you can see they got disease, okay? Here's their score, all right? Here's the germ-free animals. They don't get disease. They don't get disease because they can't mount a Th17 response. But then, if you take those germ-free animals and you colonize them with normal mouse bacteria from their guts, from normal mouse, look at this. They all develop EAE. So it's the fact that they're germ-free that they can't develop EAE. And once you put normal bacteria in their gut, they're able to develop EAE. How does this relate to humans? Well, I think this is an important carry-away point. So this is work that was done by Sarkis Masmanian and Sergio Baronzini at UCSF and Caltech. And what they did is they took gut flora from MS patients, and they took gut flora from healthy patients, age-matched, and they stuck it into those germ-free animals, all right? Here's the germ-free animals. You can see they really don't develop disease, very little disease. If they got bacteria and they're not colonized with normal gut flora, guess what? They don't get very much disease. But look at this. If they've been colonized with gut flora from MS patients, they get EAE. So there's something interesting and something different about the gut flora in MS patients that allows for this unusual occurrence to happen. So that's what was driving disease. So I said the immune system is a homeostatic system where there's both inflammation, which I just told you about. Now you have to balance that. You need regulation because you can't have an imbalanced immune system because we'd be in a chronic state of inflammation and we wouldn't survive very long because we'd lose our gut, we'd lose our kidneys, whatever organ that the immune system is involved with, we'd have problems with. So you have to be able to regulate those inflammatory cells, and that's done through the regulatory component of the immune system. And this is just a sort of a cartoon diagram of what could be happening. So if animals have too many inflammatory cells and in just the right amount of regulatory cells, they would get inflammation. Or it could be that you don't have enough regulatory cells, but you have the same amount of inflammatory cells. This is what we think happens in MS. Because as far as we can tell, in MS patients, that at least in their circulation, and if we look at their peripheral blood, their circulating immune system is no different than normal individuals, for the most part, except for during attacks. You may see a slight rise in IL-17 cells, but again, it's not overwhelming. What we do know is that the T regulatory cells and probably the B regulatory cells, remember the adaptive system has T cells, and I didn't talk at all yet about the B cells, but the B cells are the other arm of the lymphocytes that are involved in the adaptive system. It's most likely due to an insufficiency of those cells. And again, as I said before, it's not that you don't have enough of those cells, it's that they can't suppress the IL-17 response. And we think that that's what's going on for the most part. So if we take an animal that we develop and produce EAE, and these are these little black B6, they're called B6 mice, and we treat them with oral antibiotics. It's a cocktail of oral antibiotics. And then a few days later, we induce EAE, and that's, as I said before, that's one of the things that we can do is induce EAE in animals. You can see that the animals that got the oral antibiotics don't have much EAE, whereas the animals that got the same antibiotics intraperitoneally do get EAE, and it's about the same as the control animals. So oral antibiotics appear to be able to reduce the activity of these mice that are susceptible to disease so they don't have disease. Does that correlate with human disease at all? And the only information that we have at this point is an important study that comes out of Canada, and it was a minocycline trial. So minocycline is an oral tetracycline. And what they were able to show is that the patients with MS who were on minocycline, when they were on minocycline, they had reduction in their MRI activity and they had reduction in their relapse rate. When they went back to a normal off antibiotic, within about a month or so, the MRI activity returned and the relapse rate returned. So while they were on antibiotics, it appeared that there was a reduction. And it's a clue. Again, we're in very early stages of this, but it's a clue. It's the tip of the iceberg to say that, well, maybe we can adjust the gut bacteria and get to the same point with patients in MS as we do with some of these drugs that we have that are now being used. So we had the opportunity, and I'll just talk about this briefly for a moment, to use a molecule that's derived from the major commensal in our gut, which is called Bacteroides fragilis. And this molecule is called PSA. It has nothing to do with the prostate. Those of you who get their prostate measured, it stands for polysaccharide A. It's a sugar instead of a peptide. And what we found is that if we treat mice before we induce disease, we get pretty close to perfect protection against the disease process, okay? So these are untreated animals. These are antibiotic-treated animals. Or I'm sorry, PSA-treated animals. And we also know that this works therapeutically as well. So this is a molecule derived from a major bacteria of the gut. It makes up about 1% to 2% of the human bacteria. Doesn't sound like a lot, but if you got 100 trillion bacteria, it's a boatload of bacteria that you got in there. And this molecule is able to protect against EAE, both prophylactically and therapeutically. So we wanted to also look to see what happens with human cells with this. And just to make a long story short, when we looked at the mice, what in fact this molecule from the gut does is it drives a regulatory T cell response. So we could take those regulatory T cells from the PSA-treated animals and put them into normal animals, and we get protection. Okay, so this is a gut bacteria that's driving a regulatory response that is protective. So how does this change and become of interest in humans? Well, if you look at what little is known at this point, and this is primarily from two papers that were published last summer, one out of Partners at Harvard and the other one out of the Mayo Clinic, and they were looking at the colonization patterns in fecal samples from MS patients versus normal healthy controls. And what it shows is basically what we've been showing over the last 5 or 10 years in mice is that there are differences in the bacterial colonization pattern that can account, or perhaps account, again, it's a small number, for what's going on in MS patients. So if you look, the things that you look at diversity, as I said before. Diversity is the range of bacteria, okay? And all humans have pretty much the same diversity. And the first thing that they found was that there was really no difference in the diversity, range of bacteria between MS patients and age-matched controls, which is not surprising because if you're living here in Kirkland, your bacteria colonization pattern is probably not much different than people who live across the lake in Seattle, or people who live in Dallas, well, it depends on what happens with the hurricane, or with people who live in Japan or anywhere else in the world, because we're all humans and we all have pretty much the same genetic structure, and we all came from the same mother at some point. And so the diversity doesn't change that much from human to human. The other thing that you measure are abundances. Abundances is those individual bacteria that we can measure, which is not all of them, how much of them there are. And this is where we find differences. And that's where each one of us who has a fingerprint, you also have a fingerprint in your gut. So everybody's colonization pattern as far as abundances is different from one another. And even if you're twins, it's closer, but it's still going to be different. The one that's closest to you is your mom, if you're born vaginally. And that's because you come down that birth canal and you've gotten initially colonized by the same bacteria as your mom, and then that leads to what happens after that. So what they found, and you can see here, is that In the MS patients, they have a decrease of bacteroidetes, and they have an increase in the firmicutes. Is that what's driving the disease? We don't know. It's a small number of patients, so it's hard to tell. Now, if you take MS patients and you treat them with that PSA molecule that I was telling you about, what we find is that their cells go up in response to PSA, and these are regulatory cells. And I'll just skip through that because we're getting close on time here. Okay, so let's talk about risk factors in MS, and this is where we can talk briefly about the factors that are involved. Let's focus for the time being on obesity and diet. So what we know about this, and we know quite a bit about it, is that diet does change the metabolites that are being produced by the gut bacteria. So what you see here are people that have been put on either on a plant-based diet or an animal-based diet. And even after 5 days, you can see that the folks that were put on a plant-based diet have an increased amount of these molecules that lead to those short-chain fatty acids that are associated with the induction of regulatory T cells. The folks on the meat diet, okay, or the animal-based diet, I don't know if it was meat, I think it was meat and turkey. No, maybe it was just red meat, I don't remember. Anyways, they have an increase in those metabolites that are associated with the induction of those inflammatory cells, okay? This is not directly related to MS patients at this point, but this is just an observation that's made on how the gut bacteria change in response to diet. But you don't have to be a genius to carry that over and say, well, how is this going to affect me if I've got MS and the MS is due to an imbalance of the inflammatory and regulatory cells. So there's a long-term impact of diet, and especially over a year period of time. And again, there's been a number of studies, and these studies are not in backwater journals. These are studies that are in Cell, Nature, Science, the top scientific journals of the world. So that they're very rigorously reviewed for authenticity. Anyways, what these studies generally show is that people who eat a lot of carbohydrates, there is an increase in a certain type of bacteria called Prevotella, and people who eat a lot of fibers and plant-derived products have an increase in those bacteroidetes. Again, long-term diet does seem to change the microflora. And it's a stable change, too. So it's not that when you come off those diets, you go immediately back to where you were. If you go off it for a long enough period of time and go back to what you were eating before, over a period of time, it'll go back to where it was when you were 15. So the diet can change things in a variety of ways. And again, it pretty much leads to this condition of dysbiosis. So that there's a direct change of the foods we're eating on the bacteria. And it can also, as I've been talking to you about for the last 40 minutes, it can either drive a pro-inflammatory or it can drive an anti-inflammatory. So these dietary products are really important in that. As I said initially, there's a change in the intestinal permeability. Again, this is important because there was a recent paper that was first presented at ECTRIMS last year that suggests that MS patients have a leaky gut. So not only do they have a leaky brain, but they also have a leaky gut. We don't know that for sure. It's still preliminary, but it may turn out that MS is a leaky gut syndrome, just like Crohn's disease and any other disease that's associated with leaky gut, including autism. So these are the inflammatory factors that we know in diet. Again, as Dr. Brown correctly pointed out, we don't know what the right diet in MS is, but we know what diets do affect the gut microbiome, and you have to draw your own correlations at this point because we don't have complete data to say yes, 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 no. All right, so we know that foods that preferentially drive an inflammatory process are red meat, eggs, polysaturated, unsaturated and saturated fats, trans-unsaturated fatty acids, high fructose corn syrup, milk-derived fats. Why milk? We're the only species that drinks another animal's milk. A little strange when you think about it, but we do that. I love yogurt, so does everybody else. And salt. And Dr. Brown alluded to the salt being a possibility in diet that may contribute to the inflammatory process. So if you take this into consideration when you go to the grocery store and you think about what you're buying, if you look on the back of the package, which nowadays one should, you'd be amazed how much high fructose corn syrup is in all the food that we eat. And you'll be floored at the amount of salt, okay? And it may be salt in the forms of preservatives, but there's salt in there. So we're loaded with salt and we're loaded with sugar in our diet. And maybe that's all part of globalization. So we've been talking about those foods that can drive inflammation. How about those foods that can regulate? So the anti-inflammatory factors is that there's very specific receptors that are associated with the immune response, and these are called aryl hydrocarbons. And there are specific ligands that are found in vegetables, such as broccoli and cabbage and kale. I hate kale, but I always say that, and people throw tomatoes in. I just don't like it. But it's healthy. Isn't that what your mother always tells you? It used to be spinach, but now it's kale. So anyways, there is an interaction that goes between these vegetable ligands and this receptor that drives the anti-inflammatory response. And tryptophan is also a product that's associated with that, and that's found usually in fish. Omega-3, which again is found in fish, omega-6 is found in fish. They also have this propensity to drive a regulatory response. The bottom line for all this is that they're driving these short-chain fatty acids, and these short-chain fatty acids that are driving the regulatory response. So if you eat foods that tend to drive short-chain fatty acids, then you'll have better regulation. And for those of you with MS, you want better regulation because it's those regulatory T cells that just ain't working so good. So these are the known risk factors from MS. And again, this goes back to what Dr. Brown was alluding to, that there are things that we can do. And these aren't, well, I think it's fairly comprehensive at this point. So we've talked about most of them. Was mentioned smoking, because it's definitely a no-no. If you have MS, there's no question that there's a correlation between smoking and disease activity. Gender, there's not much you can do about. Sun exposure, vitamin D insufficiency, there's obviously a lot you can do about, especially living in the Northwest. I live in the Northeast, and I can tell you all the patients that we see, they're all vitamin D deficient. So I imagine the same thing, because you only have 7 months of great cloudy weather here. We have 9 months in Vermont. We're sweating in New Mexico part of the time. Anyways, stress, as was mentioned, geographic locations we've talked about, and the hygiene hypothesis, and microbial infection, again, associated with it. Is there a common denominator for all this? And the answer is yes. The common denominator for all those risk factors is the gut microbiome. Why do I say that? Because each one of those factors is influenced by the gut microbiome, and this is well laid out in the literature. So again, put them all together along with your genetic makeup, and what do you got? You got the gut microbiome. And I have hypothesized, along with members in my lab, that the gut microbiome is the major environmental risk factor in MS, as well as other autoimmune diseases. And yes, we all take therapies that can abate the immune system, it can get the immune system under control, but there's also lots of things that each one of you can do independent that will add to it, and that's the wellness part that Dr. Brown was referring to. So in conclusion, these are just a few things to think about. The microbiome is a robust and critical organ within each of us, and I reemphasize it is an organ. It's no different than your heart, lungs, and kidneys. And there's a number of factors, environmental factors, that can disrupt that, leading to dysbiosis, dysbiosis being the imbalance of that, so that the immune system is no longer in control. And finally, that the microbiome is perhaps the greatest environmental risk factor for MS. I think the reason this is important is that we, as scientists, and you as patients and families, just want to let you know that we are on the tip. If there's a tip of an iceberg, we're on the outside layer of cells of that iceberg, okay? Because we don't know anything about this organ. And what I've told you is essentially all experimental information at this point. It's going to take years to move that into understanding what's going on in humans. But when we do that, what is within our microflora truly is a treasure trove of information of how we are going to be able to treat ourselves, not in my lifetime, hopefully in my children's lifetime, more likely in my grandchildren's lifetime, that we can treat disease by adjusting the microflora. Maybe it'll be prebiotics, plants, et cetera, foods that we eat, that stimulate those bacteria that allow for our well-being. Maybe it'll be probiotics. Everybody wants to know, should they be on probiotics? And the answer is we don't know that. I know that's going to be the question that somebody's going to ask me. We don't know which probiotics at this point, but maybe it will turn out that we will be smart enough to find the right probiotic, and maybe it'll turn out that we'll have magic bullet antibiotics directed at one or two of those 2,000 or 3,000 different species that actually is seriously increased in MS patients, so that we have a bullet and we get rid of those bacteria and the other bacteria that allow for a more balanced immune system arise. And by doing so, we won't have to treat patients with some of these really powerful immune depleting agents that are now available, that are very effective, no question about it. But maybe we'll look back in 50 years from now or 100 years from now at the same ways that we looked at bloodletting, that Benjamin Rush did, when he was signing the Constitution. That was the big thing, is that you drain people with their blood, and if they don't die, we can help them, right? But maybe that's what's going to happen in 100 years from now. We'll look back and say, we did what to people? And that's what I'm hoping, because what I think is that most important is that we find the appropriate therapies that are non-invasive, that don't alter our immune systems in such a way that it can become detrimental, and we can provide patients the opportunity to have a healthy and long life with simply taking some therapies that shift things around. This is probably the final point of this, because that's what this meeting is all about this morning, this symposium, and it really is important what you eat. And take that into consideration every time you go to the grocery store and every time you go to McDonald's. I love McDonald's. But every time you go to McDonald's or you want to get a hot dog at Five Guys, and I already told Dr. Brown I saw the Five Guys near the hotel, and that's what I'm going to get the hot dog. So when people say, if you change your diet, I say, yeah, but I still eat a hot dog. So I think you have to be realistic about this, okay? I mean, that's the bottom line. You can't, nothing is purity, because we're a heterogeneous species and we're going to respond differently to different things. And these are all the people in my lab and a few others that I don't have in here that were involved in some of the science. I just gave you a smattering of the science, but these are the folks that did it. So thank you.